Hello and welcome to the B3.2 Transport Review video. Today we're going to be reviewing all of the standards you need to know about transport. The guiding questions are: what adaptations facilitate transport of fluids in animals and plants? And what are the differences and similarities between transport in animals and plants? We're going to start with Standard 1 like we normally do. And Standard 1 is all about capillaries. So capillaries are found in animals, and these are our tiny blood vessels that are used to exchange materials between the blood and the internal or external environment. So the capillaries are all about exchanging material. When you're exchanging material, there's a couple things you want to think about. You want to think about, do you have adaptations that give you a large surface area? So whenever we're exchanging things, we want a large surface area, and we want it to be able to move through, right? You need some sort of adaptation to allow things through. So permeability is the other factor we need to be thinking about. And so for these, what we're looking at here is what are the adaptations that capillaries have for a large surface area and permeability. The first thing they have is they branch, right? So our capillaries are branched in nature, which allows them to spread out. So if you start with an artery, that artery will branch into arterioles, and then those will branch into our capillaries. So then you get your tiny little capillaries. Okay, so you get your capillaries, and these are what we're talking about. So you see all the branches, that increases the surface area. They also have really narrow diameters. You can see they're very small. These narrow diameters allow more of them to be in the same space. So over here you had one artery, and you have 3 capillaries taking up the same space as one artery. So that narrow diameter allows more of them in the same area, so you have a larger surface area. Permeability. So some capillaries, well, all capillaries have very thin walls, like 1 cell layer thick. This allows for easier movement of materials through. And some capillaries have fenestrations, which are tiny holes. And this is in areas where you need rapid movement of materials. So the fenestrations are found in areas where you need rapid movement of materials. Okay? So that is our capillaries found in animals. The next type of blood vessel you have is, we're going to compare the two. We have our arteries and our veins. Our arteries and our veins. This is Standard 2. Here's a comparison between these. You need to know the structure and the adaptation. So we're actually going to do Standards 2 and 3 at the same time. So structurally, arteries look like this, whereas veins like this. And then, so you have the outer layer. For the purposes of our class, the outer layer is the elastic layer. This is a little bit of an oversimplification, right? Because it's going to be made up of a combination of connective muscle and elastic tissue, but for our purposes, it's mostly the elastic layer. The next layer is the muscle layer. And so you can see that these are both larger, so they have larger layers, thicker layers on this side here. And then in the middle, we have the narrow lumen. And so here, you've got the elastic, the muscle, and then over here, you have a wide lumen. And so why do they have different structures? They have different structures because they have different purposes. The thing that we cannot see here is that veins have valves. And so they have valves too. That's structurally is not showing up in this picture, but if we were to look at it like this, right, they would have these, like a gap like that, and a flap that blocks it. So they have little flaps of tissue that block it. All right, so why do we care? And remember, how do you decide what to do? You draw it relative to the lumen. So veins, larger lumen, and then these are smaller relative to the lumen, whereas in the arteries, they're bigger relative to the lumen. That's how you can distinguish between the two. So like, why are they like this? So the muscle and elastic help under the high pressure of the heart beats. And so you're pushing blood from the heart into the arteries. And so it's very high pressure blood because you went boom, and then it squeezed a lot of blood in there. So it's at really high pressure. So the muscle and elastic help it maintain its shape. And then the elastic layers especially, they also help maintain pressure in between beats. So that elastic layer acts like a rubber band, and it pulls it back in to keep it nice and tight and narrow. Therefore, you don't lose pressure in between beats. The other thing that we have happening here, right, is in our veins, it's the opposite. They're much wider, so they're at lower pressure. So they're at lower pressure. And this allows for it to pool blood from the capillaries, pool/collect blood from the capillaries. They also have thin, flexible walls to allow for contraction, or for compression by muscles. So your muscles will help contract your veins by compressing them. As your muscles flex, they'll compress against it and push it. And then the veins, which have valves, the valves prevent backflow. So that is some of the different adaptations between the arteries and the veins and why. So near the heart, and then returning to the heart, so lower pressure, high pressure, etc. Okay. Great. Okay, 4, measurement of the pulse, of your pulse. So there are 2 main ways to do it. And so you can do your carotid artery in your neck, or your radial artery in your wrist, and you can count and check your pulse that way. So here versus in your neck. And you can also, these are the traditional methods, you can also do it digitally. So you know, you often see the little device they stick on your finger, and it counts it for you. Okay? So you can also digitally collect your pulse. That's standard 4. Okay, oh, we also covered standard 5 over here with the veins, excuse me. So 3 was arteries, 5 was veins, excuse me. Okay, great. So now we're on to standard 6, the last SL standard for humans. Standard 6, causes and consequences of occlusion of the coronary arteries. So your coronary arteries are the arteries that supply blood to the heart. Muscle, right? So your heart is a big muscle which uses to pump the blood, which means it needs its own supply of blood because it needs oxygen. It needs oxygen so that it can do cell respiration and keep working as the muscle does. Now, there are many causes of, and it said occlusion. The standard is occlusion. Occlusion is a fancy word that means blocking. And so this is like the buildup of plaque. And so that buildup of plaque can be caused by multiple things. And then, so obesity, diabetes, poor diet, smoking, all of these things can contribute to buildup of plaque in your arteries. And what happens then is you have the consequences. So these are all correlated with it. So there's lots of different things that can do it. So it's a correlation, not a causation. One of the main ones, right, is the amount of saturated fat is very strongly correlated with it. So even if you see a strong correlation, that doesn't necessarily prove a causal link, right? So you need to be able to do experiments to decide whether it's a causal link or not. And so you have your coronary artery, and you have like the buildup of plaque, right? And then more and more plaque is getting built up. And eventually there's some like damage, and so then a blood clot forms. And that blood clot then blocks up the rest of it. And so now no blood can flow. So the consequence, right, is typically a heart attack. And so what we have happening here is there's no oxygen going to the muscle, and so the tissue stops working, and then you run into issues. And so depending on how many of your coronary arteries are blocked, the more severe the heart attack is going to be. And so as they get more and more blocked, it increases the risk of a blood clot causing a heart attack, or just tissue death of the muscle. And so that is the causes and the consequences of occlusion or blocking of the coronary arteries. All right, now we are going to move on to the SL plant standards. So the first one is that water moves from roots to leaves. So water moves from roots to leaves because of transpiration. And so what happens is you have a leaf, right? And we learned in the gas exchange unit that that leaf has little holes in it. And the water is going to evaporate out the leaf. And as the water evaporates, it was hydrogen bonded to another water molecule. And so as this one leaves, it's going to tug on this one too, right? And it's going to create this chain of pulling. Okay? So it's creating a negative pressure or a pull. And so this is cohesion causing this pulling effect. And this is going to go all the way down into the xylem. And so it would continue down into the xylem, right? That narrow tube inside the stem, the xylem, and you would see that pull going all the way down into this continuous tube. And so you have a continuous column of water. And that's because of cohesion again. And then there's also some adhesion to the cell walls, right, that cause it to stick to that as well. So there's some adhesion happening to the walls as well to prevent it from falling backwards. Okay, and so it's moving through like capillary action, right? That is that concept of adhesion is affected by that too. So it sticks to the walls of the xylem because they're very narrow tubes. Anything else for that one? Oh, I think that's pretty good. All right, standard 8. Adaptations of the xylem. So what does it have? And so the xylem is a continuous tube like this. So there's basically, like there's these cells, but there's no barriers between them. So no barriers between the cells at the ends. So they're missing the end walls. They have no cell contents, so there's nothing to get in the way of the flow of water, right? It's all about this continuous flow. They have lignin in the walls, lignin. So lignin, in addition to the cellulose, is to reinforce the cell walls and helps give it its stable structure. The tissue is also dead, just side note, it's dead, right? There's no cell content, so it's not alive anymore. And then there are little like kind of gaps for water to enter and exit called pits. And so the xylem vessels have little pits in them which allow water in and out at the different levels. And that's, if you're in HL, that's going to be important in a second here. You're expected to be able to draw, so standard 9 and 10 are all about drawing transverse sections of a stem and a root and labeling them. And so a stem is going to look something like this. Okay, so a stem would look something like this. In the middle you have the cortex, and the cortex is like our storage tissue. And this is kind of like what the tree is made up of in the middle. Then we have our vascular bundles in a ring around the outside. This is specifically for dicots. So we have our vascular bundles. Inside the vascular bundles, you have both xylem and phloem. And once again, if you're in HL, you should understand why there's xylem and phloem are together. And so typically the phloem is on the outside, and the xylem is on the inside of the bundle. So as you go around, you'd have xylem versus phloem. And then let's see, I feel like I'm missing one of the things. Oh, the outside layer. And then on the outside, you have your epidermis layer. So the epidermis layer is like the skin, and it is for protection. Okay, so the xylem is for transport of water primarily, and the phloem is for transport of sugar. Now, the root looks similar, but same things, but it is different. Instead, you have the vascular tissue in the middle, and then the cortex on the outside. Oh, that's really bad. Oh, yes. You have the there we go. And you have the epidermal layer. Okay. So here is our vascular tissue. It's now in the center, our vascular bundle. Then we have the cortex in the middle, and now we have still the epidermis is on the outside. And here the epidermis can turn into root hairs too. So you can have little protrusions called root hairs. And so that vascular bundle typically has in the very center, it would have the xylem, and then the phloem runs alongside it. The phloem runs alongside. So xylem's in the center, and the phloem runs alongside it. So if you were to zoom in, it would look something like this. Xylem, xylem in the middle, and then phloem right there. Okay. All right, that is the end of the SS standards. Now, HL only standards. So now we're going to, let's do standards 17 and 18, and then we'll go back to the human stuff. So standard 17 is, what about the roots? Why do we have roots? And so the roots actually apply positive pressure from the bottom of the plant. So transpiration creates the negative pressure we saw in the other standards, but there's a positive pressure or a push from the roots. That push is created by active transport of solutes into the roots cells. And so you're actively transporting it into the roots. Well, those solutes are typically minerals and ions, and those are going to affect the water potential. They're going to cause water to move in, so then water will enter the roots. Because you added those in, water enters the roots through osmosis. Now remember, active transport is protein pumps. Osmosis is a type of facilitated diffusion using aquaporins. So don't forget that. That's a review from the past. And so what you have happening is the water is entering. Well, when water enters, it is incompressible, right? You cannot compress water. And so it needs to move then. And so what it will do is it will push and it will move up. And so that's why you're getting that upward motion, is because it's being pushed up from the bottom. Okay. Why do they do this? Well, one, pushing and pulling is just more efficient, right? So if you had, like, you and a friend, and the one friend was pulling on the box, and you were behind it and you were pushing, that would be more effective. But now there are certain times of year where that pull isn't happening. So this would be in the winter leading into spring when there's no leaves. Well, if there's no leaves, there's no transpiration. So when there's no or low transpiration, this causes you to have a very low pull, so you need to increase the push factor. So in the winter when there's no leaves leading into spring, and in really humid environments, because an increase in humidity lowers transpiration. And so those are the two instances you're supposed to know about when transpiration pull is not enough. Okay? Alright, the last standard you need to know for the plants is the phloem. Now, the phloem is made up of what are called sieve tubes. So the phloem is made up of sieve tubes, the xylem is made up of xylem vessel elements. So the phloem is made up of these sieve tubes and companion cells. And so they're all about moving sugar. They're all about moving sugar. And so what you have is at the top, you have your sieve tube. And so your sieve tube has sieve plates, and it has holes in it, which, let me turn it, rotate it like that. So there are holes in the end of it. There's no lignin here. These are living cells. And so since these are living cells and there's no lignin, they need to keep the plates to help reinforce the structure. But the holes in them allow for the movement of sugar. So the sugar can go through the holes. Okay, so this is a sieve tube here, and then these over here are companion cells. Our companion cells, of course, have a nucleus, and they have a ton of mitochondria. Okay, let's see. In here, though, there's inside the sieve tube, there's basically no cytoplasm or limited cytoplasm and organelles. They stay alive because they have a companion cell that's giving them all the energy they need. Now, there's one last thing, structural thing we want to talk about, is that they have these like little holes in between them, between the companion cell and the sieve tube, called plasmodesmata, to allow for sugar to move as well. Okay. Now, sugar is created in the leaf, and it needs to enter the xylem. And so the leaf is, in this case, the source. And then you're going to send it to the sink, and this is where you're storing it. And so typically this is like somewhere else in the tree where you're storing it later, and you're going to store the sugar as starch. Right? So that's their energy storage. So now the question is, how do you get it into the phloem, or the sieve tube? Well, you have to active transport it into companion cell. So you actively transport it into the companion cell. So that's going to be using a protein pump. From there, it will, so that's step one. From there, it will diffuse through the plasmodesmata, or a protein channel, into the sieve tube. This is going to cause water to enter, right? So you have more solutes in there, so now water will enter the phloem. We said water is incompressible. That will increase the pressure. And then that increase in pressure will cause water to flow. And it will flow towards the low pressure. In this case, it'll flow down. So you're going to take the sugar, you're going to load it into here, and then it will flow down. And then when it arrives down here, you have to actively transport it out. and then you do this. And this lowers the solubility. So what you've done is you've created an area of lower pressure by taking that sugar and converting it into starch. So there's low water pressure here, so that the water will push it and pull the sugar down with it. We call the sugar plus water, we call this sap. Okay? So the sap is moving because of the incompressibility of water is causing it to move towards lower pressure where there's less water. That water, the water will then go to the xylem, and the water up here is coming from the xylem, so you get this kind of like cycle of water. And this is why it's important that they're found in vascular bundles, so that they're right next to each other, so you have access to that water pretty easily. Okay? All right, now let's finish up the HL standards for humans. So the HL standards for humans. So the first couple ones we're going to start with are our capillary ones. And so what you have going on here is you had those capillaries that are like 1 cell thick, and they're branching out, and they're branching out around tissue. And so they start from arterioles to a capillary, and then from the capillary they're going to go back to venules. And the arterioles are at much higher pressure. So there's high pressure here versus low pressure over here. And what we know is that high pressure causes things to move in one direction, and low pressure causes things to move in the other direction. And so what you're doing is you're forcing fluid out here, and then you're adding fluid back here. And so the tissue fluid, right, so the fluid in the tissues is created by pushing some out from the capillary, and then it's going back in due to the change in the pressure. And this is standard 11. Now, standard 12 is all about comparing blood and plasma, blood plasma, which is the fluid, and tissue fluid. And so what you have happening here, right, is the blood plasma is bringing oxygen and sugar, and then that is going into the tissue fluid where it's going to be used, and you're going to get back CO2. And that's going to then return to the venule. Okay, so you're bringing O2 and sugar to the tissue, it's being used and turned into CO2 in cell respiration. Right? We knew that. The amino acids, you're bringing amino acids, and you're turning those into proteins, and then some of those proteins are broken down, and they're turned into urea, and then that's going to end up, or also just, we can call it waste. Right? So there's waste being created in the tissues, and then that's being sent back into the venules. The blood cells, right, are only going to be found in here, with the exception of some white blood cells will make their way into the tissue fluid to go fight some things. They're like, all right, it's my time to fight. And so they will exit as well, but your red blood cells do not exit. Let's see what else. Lipids really are only found in the blood as lipoproteins, and they're not really found in tissue fluid at all. And then, yeah, I think that's pretty much it. Now, that leads us to, what about excess fluid? Well, that's 13 is your lymph ducts. And so lymph ducts collect excess tissue fluid that doesn't make it back into the capillary. So this is the fluid that doesn't make it back in. And so if we come to our drawing up here, right, we have the tissue, and then in next to the tissue, maybe I'll draw it over here. So you have your capillary, you got the tissue, and then you have a lymph duct, right, that runs alongside it and will eventually reconnect with this. And so this lymph duct looks like this, and it has valves like this, and it has holes in the wall. Some holes in the walls. And so the lymph duct has valves to prevent backflow, and it has holes to collect the fluid. So both of those are the adaptations it has to do a good job of picking up that excess fluid that the capillaries did not pick back up again. And then this will it'll eventually return to the blood. So it'll return, the lymph fluid will be filtered and it will return to the blood eventually. That'd be bad, right, if it didn't? Otherwise, you just run out of fluid eventually. So that'd be bad. So that can't happen. All right. Now we're going to talk about hearts. So you have a type of heart, and fish have a different type of heart. You can do it with a really simple diagram. So you have like this and this. Vroom, vroom, vroom. So here we have the gills, here we have the body, and here we have the heart. We have the atria and the ventricles. And so it's like ventricle. So they only have 2 chambers because they only have 1 loop. So fish, bony fish especially, only have 1 loop. Whereas mammals have a 4-chambered heart, right? So we have chambers, and we have the double loop. So 1 loop looks like this, and then this loop looks like this. Okay. So this loop is to the body, and then back to the heart, and then this loop is to the lungs, and then back to the heart. And so what you've got is you have your double circulatory system. So this is called double circulation versus single circulation. So why do we have 4 chambers? Well, it helps if I draw the other line. There we go. Why do we have 4 chambers? It's because we have that's 2 different loops, and therefore each half of the heart is for a different loop. All right, that is standard 14. Standard 15, adaptations of the heart. So you're supposed to know all of the parts of the heart, basically. So cardiac muscle, it's special muscle, right? So there's 3 types of muscle. There's skeletal, smooth, and cardiac muscle. And cardiac muscle can contract effectively like on its own. And so what it does is it is special muscle that contracts and on its own. And how does it contract on its own? Well, it's controlled by the sinoatrial node, also known as the pacemaker. And so this is a set of cells in the right atrium that tells the heart to beat. Now, there is nervous and hormonal control that tells it to speed up or slow down, but the pacemaker, the sinoatrial node, is the pacemaker, and it tells it to beat. And then things act on the SA node to tell it to speed up or slow down, but it tells it to beat. The atria are for collecting blood from either the lungs or the body. The atrioventricular valves prevent backflow from atria, or from, I guess, prevent backflow to the atria from ventricle. So they're in between the atrium and the ventricles, and blood goes from atria to ventricle. We'll see that in standard 16. And then the semilunar valves are between the ventricles and the arteries. Prevents backflow again. Prevent backflow. Okay. The ventricles are for pumping blood to, and then they contract to body or lungs. Remember, the left side is thicker because it's sending to the whole body. All right. Okay, got that. Oh, 2 things. We already talked about one of them. The coronary arteries supply. Arteries are oxygen to the cardiac muscle. All right, so coronary arteries supply oxygen to that muscle, and then the septum separates left from right side of the heart. So it is the divider between the 2. Okay. So now, those are the parts of the heart. Now what we need to do whoops. All right. So we need to understand what's happening. So the heart beats, and so blood enters the right atrium from the vena cava, and it enters the left atrium from pulmonary veins. So the blood is entering both the atrium at the same time. Then the SA node, or the sinoatrial node, or the pacemaker, sends signal to contract. This causes both atria to contract and send blood to the ventricle through the AV valve, or the atrioventricular valve, okay, that we saw on the previous page. I'm going to abbreviate it though. So it sends it through the ventricle, and then the AV valve closes to prevent backflow. So now the blood is, and this is the same thing, right? So now the blood is in the ventricles on both sides of the heart. Then there's another signal sent, and the ventricles contract. So there's a delay, and then the ventricles contract. That delay gives it time to fill, the atria to fill. So really the delay should be here before the valves close. So there's the delay, the AV valves close, and then the ventricles contract on both sides. And so this one on the right side, it sends it to the pulmonary artery. Blood to pulmonary artery. And on this side, it's blood to the aorta. And so this one will go to the lungs, and this one will go to the heart. Oh God, the heart, the body, excuse me, the body. And then the semilunar valves will close, preventing backflow. The blood will circulate, and then we'll be back up here. It'll enter the right and the left atriums. So it'll circulate to either the lungs or the body, and then it'll come back. These are the different movements of them. And I'm realizing now that I showed this to you, and I didn't read it carefully enough because I didn't emphasize this, but you need to know this graph that was in the background that we didn't really talk about. And so you have what's called systolic and diastolic pressure. And so there's this graph that looks like this, and so you have time. And on the graph, there are some lines. And there are 3 lines. Oh good, I have 3 colors right here. And so on the bottom, we have our atrial pressure. And then we have our ventricular pressure, which looks like this. This is the ventricle, the pressure of the ventricle. And then we have the aorta pressure. Okay, and so this right here is diastole. Diastole is the first part of the contraction. So the diastole is when the... Alright, so diastole is like before the atria contract, when the blood is filling the heart. And then systole is when the ventricles are contracting. And so they're like two different phases. And you spell it S-Y-S-T-O-L. Okay. And so you can see here the ventricle gets way higher pressure than the atrium ever gets. And that's important to know. Whereas the top one, the aorta, maintains its pressure pretty much throughout. And we talked about that with the elastic tissue and the muscle layer, that is what helps it maintain that high pressure in between the heartbeats. So it goes up a little bit and then it maintains it. So the ventricles up and then down as they're contracting. And so that is it.